Hello, my name is Jeff Taylor, and we are here today at the last day of the International AIDS Society meeting in Kuala Lumpur in Malaysia. And I'm here today with Dr. Dennis Burton from the Scripps Research Institute in La Jolla, who gave an excellent plenary presentation this morning on vaccines and protecting antibodies against HIV. So thank you so much, Dr. Burton, for joining us today.
Thank you.
And perhaps you could, and briefly tell us what some of the highlights are of what's happening right now in protective antibodies and vaccine research, and where do you think they might be taking us?
Yeah, I think the highlights are that we have a lot more of such antibodies now. We've had the time to study them for a couple of years, so we're gaining a lot of information from them, and this is guiding the design of a lot of new vaccine candidates, and these have now been made, and they're about to be evaluated and tested in some in small animals, some in monkeys, and some in eventually in humans. Mm-hmm. And so we expect over the next year or two to get a lot of information on how this type of approach, this rational vaccine approach, is working. So it's a very exciting time.
Now, what have the challenges been in designing a vaccine that can combat HIV? Because obviously they've been trying for 30 years. And
right. The challenges are that, well, first of all, the classical approaches don't work, so you've got to come up with something new. And then the virus has really evolved a lot of different tricks to avoid inducing the sorts of antibodies that we'd like to induce, the protective antibodies. So there's a lot of barriers that the virus has put up. It's heavily coated in sugars, for example, provides a major barrier. It's very variable. So the types of antibodies that we induce have to be what we call broad. So they have to hit isolates and strains from all over the world. It's no use just hitting one or two. You've got to be very broad. And those dealing with those, the sugar problem, the variability problem, difficult. Present a lot of obstacles.
And so you're obviously talking about a preventive vaccine here.
Yeah.
And how close do you think we are to a trial in people?
Well, I mean, there are trials ongoing now, and there will
utilizing the new approach.
Utilizing the new approach is probably several years away. I think we'd like more information on how well they work, how the sorts of antibodies that they do induce, and how good they look. So I don't think we're really ready for maybe one or two small exploratory trials in humans, but larger scale we're some distance off yet.
And right before this meeting opened, the IAS convened a Towards the Cure symposium, looking at the cure research, and they've been looking at what they call a shock and kill approach, where they kind of induce latency and try to wake up the virus and then find therapeutic approaches. Do you see this having any applicability towards that?
I think it could. I mean, there's a lot of exploration ongoing now looking at the ability of antibodies to take care of a virus that's been reawakened, if you like. Mm-hmm. And I think that's a very interesting area and just now beginning to be explored.
Yeah, it's very intriguing.
Yeah.
Other avenues do you think that are being explored, or they need to be explored in the future?
In vaccine research, or well, I mean, I think that the combination of antibodies with cellular immunity is one of the is a major goal. There are results that have emerged in the last 2 or 3 years showing very striking results with some cellular immunogens from Louis Picker that have had quite remarkable effects, and to understand how those have worked would be very valuable, and to harness those in conjunction with antibodies might be of great advantage.
So do you foresee vaccinologists putting together cocktails in the same way the virologists did with antiretrovirals? Yeah,
I think ultimately it's likely that it may be possible to build all the different features into one or two proteins or concoctions, but I think we will be looking for combinations of different actions, different effects.
Terrific. Anything else you'd like to add about the work that you're doing, or where you see the field going in the near future?
No, just that it's a very exciting time, and you know, we're very hopeful and we're optimistic, and we'll see. I mean, I don't think we're going to hit a home run, but you know, and there's still a huge amount of graft to go. But you know, things are moving more in the right direction than they have been for some time.
Yes, it's very exciting times in both arenas, in both preventive research and also the slow search for a cure.
Yeah.
Thank you so much for joining us today.
Thank you. My pleasure. Thanks.